That's a continuation of this aspect. So we go into it is often necessary to try to host several types in order to find the one that produces the highest level of biologically active recombination protein. Differences in the processing and productivity of a particular protein can occur among different yeast strains. For example, Escherichia and Polymorpha produce a truncated version of the heterologous protein interleukin 6 IL-6, whereas A. adenovirus produces a full-length version of the protein. The construction of a wide-range yeast vector for expression in several fungal species has facilitated this trial and error process, right? And on to filamentous fungal systems. Distinct from the unicellular yeast, filamentous fungal systems are also not cellular. Microscopic fungi that produce long branching strands of cells called hyphae. This group of fungi includes the common mold genera Penicillium, Rhizopus, Trichoderma, and Aspergillus. Many species of the genera of fungi are natural resources for commercially important enzymes such as antibiotic penicillin, the cancer chemotherapeutic agent Taxol, and the cellulose degrading enzyme cellulose. And the cellulose degrading enzyme cellulase, and have also been used as cell factories for the production of recombinant proteins for food, beverage, and pulp and paper and pharmaceutical industries. Similar to yeast, filamentous fungi can grow rapidly on inexpensive media, secrete large amounts of proteins, process eukaryotic mRNA, and carry out many post-translational modifications. However, an additional advantage of using filamentous fungi as hosts for the production of mammalian proteins is the ability to add mammalian-like sugars to proteins, in contrast to yeast, which typically add sugars with high mannose content. Several factors have been constructed with appropriate transcription and translation control elements for the expression of recombinant proteins in filamentous fungi. And some of these are commercially available. To achieve high yield, multiple copies of target gene are expressed under the control of a strong promoter. Commonly used promoters include the regulatable promoter from the COLA cellobiohydrolase 1 gene, CBHI from T. reese, or the glycoamylase A gene, GLA from Aspergillus niger, or the strong constitutive promoter from the glyceraldehyde 3-phosphate dehydrogenase gene from Aspergillus nidelans. However, yields of recombinant proteins are low, owing in part to degradation by extracellulose proteases. The use of protease deficient strains and gene fusion constructs has improved the output of recombinant proteins to a limited extent. The fusion partner is often a secreted protein that can carry the target protein with it to the secretory apparatus. It may also protect the target protein from degradation. And the human ILGA6 gene was initially expressed in A. nidelans from A. niger, GLA promoter protein production was very low. However, when it was expressed from the same promoter as a fusion with secreted glycoamylase A protein, IL-6 yields 200-fold, although the yields were still too low for commercial viability, possibly due to degradation by host proteases. Expression of IL-6 from strong constitutive GPDA promoter and protease deficient must improve the protein yields, but only a mutant host that was unable to acidify the culture medium. The increased yields of recombinant protein at higher pH were attributed to reduced protease activity in the altered fungal morphology at higher pH, and fungus formed mycelial pellets of that contained more live cells. This system has been used successfully to produce other human protein, the alpha proteinase inhibitor, which blocks the activity of neutrophil stays in the lungs. Individuals deficient alpha-1 proteinase inhibitor can develop to a fatal emphysema because they are unable to prevent elastic from damaging lung tissue. Purified alpha proteinase inhibitor is used for replacement therapy. However, it is difficult to obtain sufficient quantities from blood plasma, where it is normally found, and there is a risk of transferring infectious agents with the blood product, making production in a fungal host an attractive alternative. A 1230 base pair cDNA encoding the mature human alpha proteinase inhibitor gene was cloned into an expression vector under the control of the GPDA promoter and fused to the glycoamylase A gene to facilitate the secretion of alpha-1 proteinase inhibitor. The nucleotide sequence encoding the recognition site for a specific host endoprotease was included between the alpha-1 proteinase inhibitor gene and the fusion partner for in vivo cleavage of the fusion protein and release of alpha-1 proteinase inhibitor. Expression of this construct in protease deficient non-acidifying A-Niger resulted the secretion of active stable glycosylated alpha-1 proteinase inhibitor to the culture medium, demonstrating the efficacy of using filamentous fungal hosts to produce human proteins of therapeutic value. Transformation of filamentous fungi may be achieved by a variety of methods, including one, using protoplasts that have had their cell walls removed to facilitate DNA uptake, agrobacterium-mediated transfer of a vector carrying the target gene in a manner similar to that used to transform plants, electroporation, and for ballistic transformation, which shoots the target DNA into a shell on a gold or tungsten particle. This is a table of the production of human interleukin-6 filamentous fungi, host to relevant host strain, promoter, fusion partner, yield. And the methods have been successful in filamentous or filamentous fungi, and the best strategy to transform a given species must be determined experimentally. With targeted integration of a foreign gene into the genome of the yeast, SRAVC of biocombination between homologous nucleotide sequences on the vector and in the host genome usually occurs at a high frequency. Similar approaches in filamentous fungi have been less successful, and an alternative approach exploits another recombination system found in many eukaryotes, including filamentous fungi, known as the non-homologous end-joining pathway. In this pathway, the Q protein binds to the ends of induced linear DNA, and with the DNA ligase IVXRCC4 integrates the fragment carrying the foreign gene at random sites in the fungal host chromosome. This system does not require homology between the introduced DNA and the integration site. Interestingly, when the non-homologous end-joining pathway is disrupted through mutations in Q or the DNA ligase, targeted integration through homologous recombination is improved. In sum, fungal expression systems play an important part or role in the production of heterologous proteins for research, industrial, and medical applications. However, experience has shown that no one system is able to produce an authentic version of every heterologous protein. For this and other reasons, gene expression systems that use insect or mammalian cells have been developed. You have the baculovirus insect cell expression systems. Baculoviruses are a large, diverse group of viruses that specifically infect arthropods, including many insect species, and are not infectious to other animals. During the infection cycle, two forms of baculovirus are produced. The infection is initiated by the occluded form of the virus. In this form, the viral nucleocapsids, virions, are clustered in a matrix that is made up of the protein polyhedron. The occluded virions packaged in this protein matrix are referred to as polyhedron and are protected from inactivation by environmental agents. Once the virus is taken up into the midgut of this insect, usually through ingestion of contaminated plant material, and polyhedron matrix dissolves due to the alkaline gut environment, and the virions enter midgut cells to begin the infection cycle in the nucleus within the insect midgut. The infection can spread from cell to cell as viral particles, single nucleic capsids, but or from an infected cell and infect other midgut cells. This form of the virus, known as the budding form, is not embedded in a polyhedron matrix and is not infectious to other individual insect hosts, although it can infect cultured insect cells. Plaques produced in cell cultures by a budding form baculovirus having a different morphology from the occluded form. During the late stages of the infection cycle in the insect host, about 36 to 48 hours after infection, the polyhedron protein is produced in massive quantities. It continues for 4 to 5 days until infected cells rupture and the host organism dies. Occluded virions are released and they infect the host. The promoter for the poly gene is exceptionally strong, and in transcription from this promoter can account for as much as 25% of the mRNA produced in cells infected with the virus. Moreover, the polyhedron protein is not required for virus production. Consequently, it was reasoned that replacement of the polyhedron gene with a coding sequence for a heterologous protein followed by infection of cultured insect cells will result in the production of large amounts of the heterologous protein. Furthermore, because of the similarity of post-translational modification systems between insects and mammals, it was thought that the recombinant protein would mimic closely, if not precisely, the authentic form of the original protein. On the basis of these premises, a baculovirus insect cell expression vector system was devised. Baculovirus had been highly successful as deliberate systems for introducing target genes for production of heterologous proteins in insect cells. More than 1,000 different proteins have been produced using the system, including enzymes, transport proteins, receptors, and secreted proteins for a variety of applications. We just go to the diagram. The 7.14, figure 7.14 is construct for expression and secretion of the human alpha proteinase inhibitor in the filamentous fungus, A. niger. The expression cassette includes the strong constitutive promoter GPDAP, the transcriptional terminator from the TRP gene, the cDNA encoding glycoamylase to facilitate secretion, and the coding sequence for the KEX2 recognition site for in vivo removal of the glucoamylase fusion protein by the host KEX2 and endoprotease. 7.15 shows budded A and occluded B forms for ECM and PB. During budding, nucleic capsid becomes enveloped by the membrane of an infected cell. Polyhedron consists of clusters or nucleic capsids, occluded virions, embedded in various orientations in a polyhedron matrix. Bacillovirus expression factor, the first step in the production of recombinant ACMNP, will be used to deliver the gene of interest to the insect host cell to create a transfer vector. The specific baculovirus has been used extensively as an expression vector in Autographa californica multiple nuclear polyhedrosis virus. A californica, the alfalfa looper, and over 30 other insect species are infected by ACMNPB. This virus also grows well on many insect cell lines. So time? Just got 2:30, got 3 hours left. Finish, upload. Oh. the baculovirus expression vectors, the first step in the production of a recombinant AIIMS&NEET that will be used to deliver the gene of interest into the insect host cell is to create a transfer vector. The transfer vector is an E. coli-based plasmid that carries a segment of DNA from AIIMS&NEET consisting of a polyhedron promoter region and an adjacent portion of upstream AIIMS&NEET DNA, a multiple cloning site, the polyhedron termination and polyadenylation signal regions, and an adjacent portion of downstream AIIMS&NEET DNA. The coding region of the polyhedron gene has been deleted from this block of DNA. The upstream and downstream AIIMS&NEET segments included on the transfer vector provide regions of homologous recombination with AIIMS&NEET. A gene of interest is inserted into the multiple cloning site between the polyhedron promoter and the termination sequences, and the transfer vector is propagated in E. coli. Next, insect cells in culture are co-transfected with AIIMS&NEET DNA and transfer vector carrying the clone gene. With some of the doubly transfected cells, a double crossover recombination event occurs at homologous polyhedron gene sequences on the transfer vector and the AIIMS&NEET genome, and the clone gene with the polyhedron promoter and termination regions become integrated into the AIIMS&NEET DNA. With the concomitant loss of the polyhedron gene, virions lacking the polyhedron gene produce distinct zones of cell lysis, occlusion-negative plaques from which recombinant baculovirus is isolated. DNA hybridization and polymerase chain reaction assay can be used to confirm the presence of certain recombinant baculovirus. To facilitate the detection of recombinant plaques, the E. coli LAC-Z gene, which encodes B-galactosidase, is put under the control of a baculovirus promoter. It's turned on during the early to late stages of the infection cycle, and this construct is made part of the DNA unit that is incorporated into the AIIMS&NEET genome. Recombinant plaques turn blue when a chromogenic substrate for beta-galactosidase is added to the medium. Additional rounds of infection of infected cells with viruses from occlusion-negative plaques increase the concentration of recombinant viruses. Heterologous protein is harvested 4 to 5 days after host cells are infected with a high titer recombinant baculovirus stock. Let's just read through the diagram. Organization, the expression unit of a baculovirus transfer vector, the gene of interest is inserted into multiple cloning site MCH that lies between polyhedron gene promoter, PP and polyhedron gene transcription termination PD sequences. AIIMS&NEET DNA upstream from the polyhedron promoter and downstream from the polyhedron transcription termination sequence provide sequences for integration of the expression unit by homologous recombination into AIIMS&NEET genome. This is the table of some of the recombinant proteins that have been produced by the baculovirus expression vector system. Alpha interferon, adenosine deaminase, anthrax antigen, beta amyloid precursor protein, B interferon, bovine rhodopsin, cystic fibrosis transmembrane conductance regulator, dengue virus type 1 antigen and erythropoietin, G protein coupled receptors, HIV1 and envelope protein, HSV capsid proteins, human-like alkaline phosphatase, human DNA polymerase alpha, human pancreatic lipase, influenza virus hemagglutinin, interleukin-2, Lassa virus protein, malaria proteins, mouse monoclonal antibodies, multi-drug transporter protein, poliovirus, pseudorabies virus, glycoprotein 50, rabies virus, glycoprotein, respiratory syncytial virus antigen, simian rotavirus capsid antigen, and tissue plasminogen activator. Thank you.